Let's talk about the Watson and Crick DNA model. Here's a quick overview of what I will touch on, so let's get started. In DNA, you have two kinds of nitrogenous bases, purines and pyrimidines. Purines are two-ring structures as shown on the bottom right, and pyrimidines are single-ring structures as shown on the top right. There are two kinds of purines, adenine and guanine, and two kinds of pyrimidines, cytosine and thymine. Adenine always binds to thymine using 3 hydrogen bonds, and cytosine always binds to guanine using 2 hydrogen bonds. Keep in mind, hydrogen bonding is weak. It's the weak attraction between the positive and negatively charged molecules, and it is not the main force that joins the two strands together. Hydrophobic interactions are going to be the main force stabilizing the helix. This hydrophobic interaction is maximized by base pairing, as the nitrogenous bases are hydrophobic and are stacking within our double helix. Nucleotides are composed of 3 parts: a sugar, a base, and a phosphate. The phosphate backbone is going to be the outside of this latter part here. In DNA, the sugar is deoxyribose, which is a 5-carbon sugar ring. We label each carbon clockwise. The first prime carbon is attached to the nitrogenous bases. The second prime carbon will help us differentiate between RNA and DNA. DNA will not have an OH group, but just an H on the second carbon. The 5' carbon is attached to the phosphate, and the 3' is attached to the next 5' phosphate of the next nucleotide in the sequence. Here's a better look at how the 3' attaches to the next nucleotide. The major groove in DNA is wider than the minor groove, and it occurs where the phosphate backbones are further apart from each other, while the minor groove occurs where the phosphate backbones are closer together. DNA has 2 directions, right and left-handed. The easiest way to tell is by drawing arrows to see which way the strands are pointing. Right-handed DNA is much more common than left-handed DNA and can be identified by the arrows that point up to the right. Left-handed DNA, while uncommon, is identified the same way, but with arrows that point up and to the left. The nitrogenous bases pair in a specific way, as mentioned earlier, making the strands complementary, in which they must run in opposite directions, making them anti-parallel. There are negative charges present in the phosphate backbone. Putting DNA in higher concentrations of salt solutions can partially neutralize the negative charge in the phosphate. Let's look at how that would affect the midpoint of thermal denaturation as well as the properties of DNA. DNA in high concentrations of salt would become less hydrophilic, meaning less soluble in water. The positive charge on salt would neutralize that negative phosphate, making it more stable and harder to break. This means we can conclude that DNA in higher concentrations of salt would increase its midpoint of thermal denaturation due to its increased stability. Here's a closer look at how increase in salt increases the temperature needed for denaturation. And here are my references.